Alright, good evening. Like, whatever. Whoever wants to watch this video, I guess. This is, telling people you have a base knowledge of molecular biology 101. So, you probably saw in the description thing the bit where it says all, like, bruh, I don't know what I'm doing, but I'm going to try and explain these concepts since, we're not really getting that in my class here, so I'm gonna, it helps me to have it all, explain it to someone else. It helps me study and all that, and maybe I'll help someone along the way. So, I have this, my lovely textbook here, McGraw-Hill Connect. This might be piracy, don't sue me, please. And today we're discussing the chemical basis of life 2, organic molecules. Go at you. Alright, so, we're just gonna mostly go over the stuff that's highlighted or whatever, so we don't have to do all the nitty-gritty stuff. You have to, like, actually read your textbooks, kids. I'm not a complete instructional video, but I do my best. Alrighty, so we're starting off with carbon is an essential element for life. Basically, the important thing to know about carbon here is carbon has 4 filled and 4 empty slots in its valence shell thing. So, because it has those 4 open slots, it can fit a lot of different other molecules in it, and that gives it basically a lot of different things can go into it in a lot of different ways, and that gives it a huge versatility, and that's why it makes up a huge majority of organic molecules. So, we have these lovely things called polar and nonpolar bonds, and hydrocarbons have mostly, like, nonpolar bonds, I believe. Alright, let's start with what polar versus nonpolar is. Polar basically means it's in balance. Imbalance. It means there's not a balance. It's not good. There is an imbalance in the elements. Someone call the avatar, etc. So polar elements are very susceptible to, like, water and those types of things because as a mnemonic, water thrives on chaos. You ever see an ocean, a river, and anything? There's order, but it's wild usually. And because water likes to attack polar things, and since polar is imbalanced, water goes yeah and tears it apart. So being nonpolar basically means it's balanced. The water can't really get to it. It's not usually soluble as easily. So the hydrocarbons are molecules that are largely nonpolar. And that means they're hydrophobic, which means they're afraid of water. That's what it technically means. For here, it means it's not really soluble in water. And then, so that's your hydrocarbon thing. So it has predominantly your CH and CC bonds here, which is, okay, I don't know where the highlight thing went, but that's supposed to be there. So anyway, like carbon and hydrogen bonds here, and carbon bonds. Exciting. So we can see here that we have these bonds here are all electrically neutral and nonpolar. All good, safe and sound. These are polar. They're oxygen has more electronegativity over here than carbon does, so that means it's going to be polarized. Alright, so aside from hydrocarbons, most of the organic molecules and macromolecules and stuff have functional groups, which are just these lovely groups of atoms that have your nice chemical structures and stuff. They're kind of like cliques. We have all these nice ones. We got your aminos, which act as a weak base. Sorry about all this nonsense box thing. I don't want to be there, but it is. The amino is good because it's a base, and when combined with carboxyl, which is an acid, they go together and form a peptide bond, which will come in later, but not yet. You see them a lot in amino acids. That'll come in later at like near the end of this video. But hold on to your horses, kids. We have a ways to go. So, got your carboxyl and your amino. Remember that. Acid and base. Important. So, carbonyl, whatever, is really reactive, helps form hydrogen bonds. It has these two little things here, ketone and aldehyde, which are very similar except for this one has an H, this one has an R. And that's enough to change it. You find those steroids, waxes, proteins, etc. Those are also later. Basically, these are what you might want to like write these down, reference them later because you'll see them later, and it's helpful to look back and forth and be able to get a basic understanding of what's going on here. So now we're going to go to hydroxyl. It's got some nice hydrogen bonds with water, nice and simple. Methyl is the only one out of these that's a nonpolar, which again means balanced, non-water soluble, etc. Some has attached to like DNA, proteins, carbohydrates. Nice little bond here thing. We have phosphate. Some phosphate and sulfate are both. What does it say? I have notes here, but I don't remember what they say. All right, so these both have these are both negatively charged at the typical pH. And then sulfhydryl. I think that's how it's pronounced. Anyway, it has this lovely amino acid cysteine here. It has this amino acid here. All right, and it helps and it makes these disulfide bridges in many proteins, which we'll get into later. Okay, all right. Now we're gonna scoot on down to this. Basically, hold up, I skipped something. Nope. Okay, that's all on one page. So basically, there's like, what the heck? Alright, I have a note for a later one apparently. Alright, so this is basically for all these organic molecules we're going to look at, this is how they synthesize and come together, and it's how they break down and break apart. So they come together from a dehydration reaction. Basically, you got these little monomer things here, and they got their stuff here, and they also have this lovely little water atom thing here. And when they come together, you have to first take out the water. So when the water comes out, they come together. I have this lovely illustration here thing. Imagine monomers are like introverts. They have their books, they refuse to socialize. Now these books are representing water molecules, because when you take the books away, like a mean parent who's always saying, go socialize, they have no choice but to all go hang out together. This creates a polymer. Da 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 da. Now that is in a dehydration reaction. In a hydrolysis reaction, you take all those lovely introverted monomers and give them their water books back. And now they have no reason to socialize. They are happy to be back in their own worlds. They are no longer a polymer, they are now a monomer. For the future reference, mono is 1, di is 2, poly is when everyone got lazy and just decided that that was good enough for anything above 2. So now I got this lovely page here, which basically just all this nonsense to say there's 4 categories of organic molecules and macromolecules: carbohydrates, lipids, proteins, nucleic acids. And we're going to get into each of those in detail now. Starting with carbohydrates. Basically, these are, fun little fact here, carbon, hydrogen, oxygen. You take the carbon and put an O for oxygen, and then put the hydrogen, you get carbon, oxygen, hydrogen, carbohydrate. Alright, well, that only kind of works because it's basically the same for all of them. Alright, anyway. These are all things that are sugars broken down to sugars, etc. You're going to see most of these names on, like, the back of, like, candy and stuff. Because they're sugars. Good old sugar times. Alright, so basically, all these little carbon things, they can form these lovely monomers, and these are called monosaccharides, which is Greek, means simple sugar. So the most common one is called pentoses. Pent means 5, like pentagon, if you learned basic shapes. So they have 5 carbons, which is pentoses, or 6 carbons, which is hexoses, like a hexagon. Whoa! Shapes! Who knew they'd be useful? Anyway. Let's see. So those are, like, examples of monosaccharides. I think these are all, these are all hexoses. You can tell because 1, 2, 3, 4, 5, 6. All those little numbers, those are the carbon numbers. Each of these is going to have, their carbons numbered. So even though you can't see it exactly, that's basically where there's 6 carbons all together. And that makes the numbers. The numbers connect. When certain numbers have with certain other numbers, they do cool things like make branches and all that stuff, but that's for later. For now, monosaccharides are pentoses or hexoses. Now, if you take those lovely things and stick them together, they're usually a dehydration reaction thing. They combine and become a disaccharide. I think that's right. Anyway, yeah, they're a dehydration reaction. You can take 2 of these lovely monomers and turn them into monosaccharides and turn them into a disaccharide. And then, of course, they lose their water because they're shaking their books away and saying, stop that, go socialize. So, when you have lots of monosaccharides, they make a polysaccharide, just like that thing we were talking about. So, like, polysaccharides are like starches, glycogen, cellulose, pep, that, chitin, that. So here's an example of starch. These are all, what is this called? Alright, so these are all glucose, all these little squares. Now when this first carbon and this 4th carbon come together, it makes a chain. That's how it works for glucose. This different numbers for different monosaccharide things, but in this case, a 1 and a 4 makes a chain, and a 1 and a 6 makes a branch. And the difference between starch and glycogen and cellulose is just whether they form in branches or chains. That's all. See? Branches. Voila. Chains, branches, a little bit of each, but a little more branchy things. Sorry, chain. A little more chain things. It's a starch. Cellulose, it's all just changed. That's all. So that's how it's all different. That's not very complicated when it gets down to it. Let's see. But yeah, like starch and glycogen, that's used for energy. Like if you don't have energy, basically, it's for storing. You may have lots of energy now, so your body's like, aha, I'll go put this in the frickin' starch or glycogen containers or whatever. And then later, when you're like, man, I could really use some energy, your body's like, oh, great, I have this stored up. That's kind of how it works. So, like, yeah, fun times. So, those are carbohydrates. Carbohydrates, the sugars, that sort of thing. Next up is lipids. Now, these lovely lipids are hydrophobic, for one thing. They don't like that water. They're nonpolar and don't dissolve in water. So there's like kinds of lipids are like triglycerides, which are like fats and oils, that sort of thing. And then you have waxes, which, so you know about wax, like on crayons. You ever made those wacky things in art where you just take your crayon and stick them all over the place, and you put your watercolor all over it, and none of the crayon gets touched, as long as you did it right, by the watercolor, because watercolor is water and doesn't want to touch that wax on that crayon. So you just have your lovely design thing here, and there's no, it doesn't get washed away. Graphic design is my passion. Alright, so here we see glycerol and 3 fatty acids. Take away their water through dehydration, and it makes triglyceride. These lovely little bonds called ester bonds. So here we have, like, wait a moment. Yes. So, saturated fats, saturated fatty acids, just have these nice single bonds here, all just nice and in a row. And since they're so in a row as compared to these wild fools, they can stack together, and you can get lots of them stuck together really easily. So it makes it makes it very hard to break. So if you have like hamburger grease or something like that, it'll harden in normal temperature. When you put it in the pan, it'll break up because you're putting heat in it. But at room temperature, it's going to be hard. So that's an example of a saturated fatty acid. An unsaturated fatty acid is like that. It has these little double bonds that just give it this bendy thing. Now these are kind of all over the place, and they're not as tightly packed, which means they're usually flopping all over the place. Oils. Oils are at room temperature. Oils are unsaturated fatty acids. Let's see. And as you may have guessed, a monounsaturated, so there's saturated fatty acids, they're single bonds. Monounsaturated fatty acids is when you have one little double, and that makes it the weird bendy shape. But polyunsaturated is when there's 2 or more double bonds. So they just keep going, and it's all over the place. So those are saturated, monounsaturated, and polyunsaturated fatty acids. Now we get to phospholipids. Phospholipids are almost the same as triglycerides, but instead of getting 3 fatty acids with the glycerol, it takes glycerol, 2 fatty acids, and a phosphate. Now, the fatty acids here don't want to be in water, but the phosphate thinks water's pretty cool, which basically ends up like this. All your phosphates are your polar heads. They are polar, so they point outwards towards the water. They think it's pretty great. And then all the fatty acids turn inwards to get away from it, and they can form this nice long chain of polar and nonpolar. And when it's both hydrophilic and hydrophobic, it's called amphipathic. Da 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 da. So that's a phospholipid. Amphipathic. That's the important part. Now we have steroids. Steroids, basically, the only special thing about them, is they got these 4 rings. 1, 2, 3, 4. And they're used for fungi, plants, animals, makes cholesterol, estrogen, all that fun stuff. Steroids, nothing wild. And then waxes are lipids that prevent water loss. And like we discussed, it keeps the water away. So that's a lipid. Lipids are fats, carbohydrates are sugars, and then you got your proteins. Proteins are kind of complex because proteins are made out of amino acids. And amino acids all combined together make, hang on, it's all written down. I'm going to give you this overview right at the start. Amino acids all line up and make a polypeptide. Polypeptides do all kinds of twisty stuff and turn into a protein. I'm pretty sure I misspelled protein, but don't. That's not important. Do you want an education or no? Alright. So, amino acid, polypeptide, protein. So let's talk about that. So, start with the amino acid. The amino acid has an alpha carbon right here. That little A thing is an alpha. Looks just like an A because that's where it comes from. Ah! Get out of here. I did not ask. No, stop. Okay. So there's your alpha carbon here, your amino group, and your carboxyl group. Amino and carboxyl group, you say? Wasn't that the thing we were looking at way back? Oh, it's not that one. Wasn't that the thing we were talking about way back? Way back here? Why, yes, amino and carboxyl. That is the same one. I'm so glad you wrote that down and remembered. Alright. Welcome back to losing my pillies. Alright. So your amino group, positively charged at neutral pH. Carboxyl group, negatively charged at neutral pH. Alpha carbon. And then this is the amino acid side chain. And this is what's different in each and every single amino acid. And here you can see that. So we start with the alpha carbon. And then all the other stuff. Let's see. Yeah, alpha carbon. There's your carboxyl group. There's your amino group. There's your little hydrogen thing. And here's the different side chains. And each one of these is different. Blah, 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 blah. These are polar charged. These are polar uncharged. These are nonpolar. But the main thing to focus on is that the side chain is different, but carbon, amino, and a carboxyl, same. That part's the same. Got it? Great. So, the thing about amino acids is that they all have to line up eventually. In something like this. Each of these little dot things is an amino acid. They all line up in a nice primary chain. And that primary chain is formed because you got your alpha carbon, your amino group, your carboxyl group, and your side chain. Now on your amino group, there's an N terminus. On your carboxyl group, there's a C terminus. And N terminus and C terminus combine. So this amino acid can combine to this other amino acid by the C terminus and N terminus. And whatever. And when the C's and N's, C, N, C, N, C, N, they all line up into this nice primary chain thing. And that is the start of a peptide bond. We heard about peptide bonds, didn't we, kids? So that means this big, wacky, crazy thing is basically summed up into alpha carbon, side chain, amino group, carboxyl group, and the neighboring amino group together. Amazing. So they're combined here into a peptide group. And then here's another amino acid, and another, and another, and another, and another, and another, and another. and there's the free end. So each of these, though wild and crazy as it seems, is just one of these, combined to one of those, combined to one of those. So all you have to remember is that C-terminus and N-terminus go together. Make this nice chain. Got it? Great. Look at that. Octopeptide. We call them polypeptides because we're lazy. So, here's where it gets a little complicated. So you take this lovely primary structure. We finally have it all figured out. It's all just these lovely little alpha carbons, hydrogen, side chain, amino group, carboxyl group, stack, 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 stack. But now it has to get complicated because they have to attach in either an alpha helix or a B pleated sheet. And these go together because let me see if I remember why they do it that way. Okay, so we're getting to that part later. For now, remember primary, then it's told to go into either an alpha helix or a B pleated sheet. And from there, those lovely things are all scooshed together into a tertiary structure? Tertiary? Into the third step here. And these all stick together in different ways. And that becomes this. Now, most proteins will just stay here, but some are complicated enough that it goes to the quaternary, maybe, something like that. It's where these structures all come together. You see one here, one here, one here. And that becomes a more complex one. It's a hierarchy. So, primary, that turns either an alpha or a beta. And then a bunch of those secondaries get stuck together into a tertiary thing and become tertiary. Then you take the tertiary thing and stick them all together. See, I even use the same one so you can see it clearly. And become a quaternary. Do I know how to pronounce any of this? Absolutely not. Okay. So remember how we got to this? Amino acid all lines up with those wacky bonds here. Why? They turn into a polypeptide. The polypeptides do some twisties, and they turn into a protein. Protein. It's there, okay? So the way that they know to become a helix or a pleated sheet, or then to turn into this, and then to turn into this, is through protein structure, which is formed from hydrogen bonds. So weak hydrogen bonds act to a collectively strong force that promotes folding stability. Ionic bonds and polar interactions. Some side chains are positively or negatively charged. The positively side chains may bind to negatively charged side chains. 1 second. Gotta water the old throat. The positive goes to negative. It makes an ionic bond. Hey, it's starting to come together. Hydrophobic effect. Some amino acid chains are nonpolar. As the protein folds, the hydrophobic will run to the center where they can't get hurt. And the hydrophilic will run to the outside so they can go greet the lovely water. And since some parts are going in and some parts are going out, we're starting to get a structure. And then van der Waals dispersion forces. So atoms within molecules have temporary attractions if they're optimally distance apart. So kind of like magnets, like they don't really work if they're like across the room, but if they're close to each other within like a couple inches, they'll zip together. That helps with tertiary and quaternary. Whatever. And then disulfide bridges. The side chain of the amino acid cysteine. I don't know how to pronounce any of this. Anyway, the cysteine contains the sulfohydryl group. I'm sorry, this stupid mouse. which, when reacts with another group, another cysteine side chain creates a disulfide bridge, which links them together. And they're covalent bonds that occurs on a polypeptide or between different polypeptides. Now you may remember way back up in, not this again. This thing. I asked for very little. When we discussed sulfhydryl, when I said it creates a disulfide bridge, that is this bridge. And here's cysteine. Haha, cysteine, very nice. So that cysteine, I went past it. That cysteine has the lovely sulfhydryl. The sulfhydryl sticks together, makes a disulfide bridge. Cool and beans. Now this here is just this nice experiment that basically says that proteins know how to fold, and even if you break all their bonds, once you take out the bond breaker juice, it just goes back together. Wild. And then this bit here just says it's got its little functional domain thing. It's got, yeah, like the same kind of domain. I don't really get this part. Basically, it's got this little bit, and that's what makes it unique. It can look exactly the same, but this little thing right here is what changes it. Alright, so now we come to nucleic acids. Now, you've probably heard of nucleic acids because they come in deoxyribonucleic acid, or DNA, or ribonucleic acid, or RNA. See, you can call it deoxyribonucleic acid if you want, but you'll sound like Jimmy Neutron. Alright? So, basically, DNA and RNA also have polymers made of monomers, except each monomer is called a nucleotide. And each nucleotide has a phosphate group, a pentose sugar, which is either deoxyribose, which sounds awfully familiar, or ribose. Deoxyribose for DNA, and ribose for RNA. So phosphate group, pentose sugar, one for each, and then a single or double ring of carbon and nitrogen atoms known as a base. So here we can see it here. You got your phosphate. You got frickin'. Alright, 1 second. Alright, so you got your phosphate. You got your, I believe this is deoxyribose. And then base, maybe. Bruh, I don't remember. Let's stick with, there's phosphate, pentose, and carbon and nitrogen base thing. Alright, so, and if you remember way back when I was talking about, I'm sorry, I'm doing it again, carbohydrates. Carbohydrates, and we said that 1s and 4s connected and became chains, and 1s and 6 became branches, and I said that that's not the same for all of them. Here we can see that 3s and 5s combine via phosphate group, but in this case it's 3 and 5, not 1 and 4, or not 1 and 6. But this is getting a little wild. Phosphate, pentose, single ring or double ring of carbon and nitrogen atoms known as a base. Okay? So, now we're going to talk about DNA specifically. DNA uses deoxyribose as its pentose sugar. That's how it gets its name. Now the thing about deoxyribose is it has 4 different nucleotides that can link to deoxyribose. Those are then divided into two different categories. The purine bases and the pyrimidine bases, which are then divided into adenine and guanine for purine, and cytosine and thymine for pyrimidine. So that's complicated. So A and G for purine, C and T for pyrimidine. It's also worth noting the adenine and guanine and the purine thing have double rings of carbon and nitrogen atoms. See? Double rings. So, yes, this is the phosphate, this is the pentose sugar, in this case deoxyribose, and this is the base, which is a double ring of carbon and nitrogen. In this case, no, I'm not even going to try and go there. That's the nucleotide part. So here you got your purine base, and here are your pyrimidine base. You can see the 2, see the 1, all cool. And then it gets slightly more complicated yet again. Bro, a cat. What's up, cat? Alright, well, unless the cat wants to watch. Now we get to the part where the nucleotides start coiling to form a double helix, and it gets slightly more complicated because the purine and the pyrimidine only go on opposite sides. So a purine has to connect to a pyrimidine, and specifically, adenine connects to thymine, and guanine connects to cytosine, whatever. So G and C go together, and T and A go together. I guess I can help you remember, like, the little curly ones, and those go together, and the other two go that way. So, let's recap again. A and T, G and C. Purine, pyrimidine. Nucleotide strands, DNA. Sweet. So that is DNA. Now, I found my other note. Here it is. And we can discuss RNA, which is very, very similar, except it has its phosphate. Its sugar thing is ribose instead of deoxyribose. And then when it comes to the whole A and T, G and C thing, the A goes with the U, because instead of thymine, it has uracil. But adenine, guanine, and cytosine, all the same. So that means that A and U go together for RNA, and C and G go together for both. Yeah, that's a lot, but if we recap, yet again, we have carbon. Carbon is extraordinarily versatile. It's got all kinds of fun groups that bond to carbon. And when these fun groups go and wander off together through dehydration and hydrolysis, they are revealed as carbohydrates. They're sugars, lipids, which are fats, proteins, which is energy mostly, and nucleic acids, which is everything else. Okay, one thing I forgot to cover. When it comes to nucleic acids, DNA basically encodes the genes. It stores all the genetic information, and then RNA decodes it. It tells everything else how to turn all those amino acids into proteins and all that fun stuff. So, I think that about covers this chapter thing here. You can hopefully now say you have a basic idea of whatever the frick molecular biology is. You can kind of see, like, here's those little twisty A helix structure things, and I guess, maybe. Did I actually say what this is? Yeah, a model showing two proteins that have been bonded together. And what does that make it? That word I can't pronounce. Well, alright, I'm gonna just stop. Hopefully that made, like, a lick of sense. Perhaps even 2 licks of sense. And in the extremely rare cases, maybe it was enough licks to get to the center of your Tootsie Pop, and you can now pass this class. Toodaloo, serfs. This is Queen Elizabeth pointing at science. Signing off. That was really cringe.